Let's kick today's session off. For those who don't know me, my name is Helen Dallas from the Freshwater Research Centre, and together with my colleague Jeremy Shelton, we are your hosts or your trainers for the 9th FBIS training session. So great to see many familiar names here, and as always, a big welcome and thank you for your interest in FBIS. Okay, so this session is being recorded and we will share it afterwards. But for now, what I'd like to do is just open up the schedule of our training just to remind us where we all are. So I know several of you have actually been at all of the training, which is great. But where we are, we started in September and we're all the way down our second last session for this year, session 9, where we'll be really diving into adding biodiversity data. And we have a pre-recorded tutorial on this, which we'll start in a minute. And then next week, we'll be doing our last session, which is adding SAS data. So these are probably last session on adding sites and adding data and adding SAS data are critically important because the whole purpose of the training that we're doing is to try and encourage you, the data providers and the freshwater community, to add your data so that, you know, it adds to the bigger system of FBIS and is available not for a resource for everybody to use. So it's all good and well using the data from FBIS, but we really do want to encourage adding data and data providers to use it as their vehicle for curating and consolidating any data, be it a thesis from your master's or SAS data, etc., etc. So as always, we always try and encourage the becoming part of the freshwater community and FBIS community and embracing FBIS as your vehicle for not only using data, but storing your own data going forward. Okay, so I'm always happy to take questions from previous sessions if there are any before kicking off with today's session. Are there any specific issues? Just shout out if there are. I'll give you a few seconds. No? Okay, so I think what I'd like to do then is to jump over to the pre-recorded session about adding biodiversity data. As always, please either jot down your questions as you go along and we can have a conversation afterwards, or type your questions in the chat. We've got an hour together, so let's maximize the time that we have. Okay, so I'm going to just stop my video and put it onto the recording. Excellent, enjoy.
Session number 9. In the previous session, we went through the workflow of creating a new site.
Sorry, I'm just going to pause this quickly because the volume on my side is not very good and I'm sure that there's going to be an issue. I'm just going to stop share for a second. Sorry, one moment. Bear with me. I want to just increase the volume. I can hear that it's not particularly strong today. Almost there. Okay, let me up that. Share my screen again. And let's try again.
Greetings, everybody, and welcome to FBIS training session number 9. In the previous session, we went through the workflow of creating a new site on FBIS, and today our focus is going to be on adding biodiversity data to a site, to an existing site. Right, so there are different ways that you can access the site on FBIS. The one way is to enter the site code into the search bar, but you can also just navigate the map and identify the existing site to which you would like to add biodiversity data. All right, so we're going to zoom in on an area a little bit to the north of Worcester in the Western Cape, and we're going to add some new biodiversity data to an existing site on the Suntriff River. You can see that there are already existing invertebrate data for this site, but no other biodiversity data so far. All right, so the way that you access the data upload form is by clicking on this little add button, and there's a separate form for each biodiversity module. So this would be the one for fish, invertebrates, algae, adult odonates, and anurans. You can also add SAS data using this button and physicochemical data using this button over here. But these are two separate workflows that are going to be covered in upcoming tutorials. All right, so for the case of adding new fish records to this existing site, we would hit the add button, and that takes us into the fish data upload form. The top of the form is a map showing the location of the site, and then the spatial coordinates of that site. You then have an option to enter the date that your sampling was conducted, and the owner is auto-populated as myself because I'm currently signed into FBIS. But if I was uploading data on behalf of somebody else who actually collected the data, then I would enter their name. Next up is some information about biotope and substrate. So for fish, there are 4 different kinds of broad biotopes. These are different combinations of flow and depth, commonly used in fish sampling. In my case, it's the slow deep habitat that was sampled, but if you weren't sure, you can also indicate the mixed habitat. For the specific biotope, my sampling was conducted in a pool, but there's quite a range of different specific biotopes available here. And the substratum, well, that's really the dominant substrate type at the site where the sampling was conducted. Then you need to specify the sampling method that was used to collect the data. Now, just to point out here, not all occurrence records for fish or for any other freshwater biodiversity necessarily have this information, but if you did collect information on sampling method and effort, this is where you can enter that information. So there's a drop-down menu including all the most commonly used sampling methods for fish, right the way through from electrofishing down to underwater video analysis. In my case, however, samples collected using snorkeling. Then you can specify the effort, and this could be specified using either time or area. In my case, the snorkeling was conducted for 15 minutes in the pool. Then you can specify the record type. The options here are visual observation, photographic record, specimen collection, acoustic record, most commonly used for anurans, DNA sample, which also includes eDNA as well. So since I was doing snorkeling, the record type would be visual observation. You can then upload an image of your sampling site if you like, and that will let you navigate to wherever you have saved your image of your site. In my case, it's on my desktop, so I can upload that image file. And then now that we've added the basic information about the sampling, how the sampling was conducted and what the habitats were like, we can then go on to add the actual fish biodiversity data. So here we have a few columns. The first column is the taxa, and then whether or not the taxon was observed, and then the abundance of that taxon, which can be indicated by number, by percentage abundance, or by density. The list of taxa auto-populated by FBIS includes taxa that occur within the general area where the sampling site is located. So it's a nice kind of short list of likely taxa that will be included in your sample, but not necessarily so. So in my snorkel sample, we did record one non-native species, the rainbow trout, Oncorhynchus micus. And so then I would tick that that species was observed. And I know that we saw 3 rainbow trout in that snorkel survey. But we also recorded some native species, the Breda River redfin, of which we saw 7, the Cape Kerper, which we saw 3, and last we saw Cape Galaxius, and we saw a nice big school of 10 Cape Galaxius. So we've entered the taxa that we observed and their abundance measures, and now we're ready to submit this data. And the last step before uploading these fish data is agreeing to the FBIS terms and conditions, which is that the data will be shared via the FBIS platform for visualization and download by other FBIS users. Right. And then a pop-up comes to confirm your submission. All right, then the next form is for abiotic data that may be associated with your biological sample records. So once again, these data are not always available and were not always collected alongside the biotic data, but in cases that they were, this is the form where these data can be uploaded. Abiotic parameters associated with those biological sample records. And just to give you a quick overview of this, because there's going to be another session focusing on the details of adding abiotic data, you have options to specify some basic parameters about the habitat in the top here, including the water surface width, the width of the wetted channel in categories, the water level, an indication of flow, the water turbidity, a basic indication of water clarity, how much canopy cover there was, and then the embeddedness of the substrate particles. Then there are a whole range of different parameters that are often also collected alongside biodiversity data. And the first one is alkalinity. And you can just indicate using the checkbox whether or not you measured any of these, and then you can just include the value for that parameter. And this includes lots of different options, all the way through from different nutrients to things like dissolved oxygen and pH, conductivity, really nice comprehensive list. Yeah, and then just like for the fish form, once you're happy with those data, you would hit agree and move on to the submission. And the last step in uploading your records is to specify the reference category associated with these data. Now, the different reference categories are database or peer-reviewed scientific article, published report or thesis, or unpublished data. And this may be a reference that's already on the FBIS platform, or you may be wanting to add a new one. In my case, it's a database, and the database is called the FRC monitoring database. But if your reference did not appear in the existing ones on FBIS, then you have an option to add your new reference for those records. All right. Next up, you can add any notes about the about the sample collection. In my case, it was water was very cold, fish inactive, indicating that I may not have seen all the fish present in the pool. And then I would submit, and that is really the final step of uploading your biodiversity data to FBIS. Submit. All right. So now what I'd like to do is just go back to the map and point out a few other differences that can be found when uploading data for some of the other biodiversity modules. So if we navigate back to our site, just north of Worcester, we'll be able to see our new fish data have appeared there, which is great. Our upload worked. And what if then we wanted to add some more invertebrate data? We would hit the add button here, and that would take us to the invertebrate data upload form. Now, I'm not going to walk you through everything because for the most part, it's very similar to the fish upload form. There are a few subtle differences that I'd like to point out. In terms of the broad biotopes, if you remember for the fish, we had those 4 depth flow categories, but in the case of invertebrates, there are some different options, including stones in current and out of current, marginal vegetation, aquatic vegetation, GSM, and the list of specific biotopes is similar, as are the substrata. And so the sampling methods are also unique to invertebrates. If you remember, for fish, we had things like electrofishing and snorkel sampling. For the invertebrates, there are other kinds of methods like server sampler or kicknet or light trap that are that cover invertebrate sampling in general. And otherwise, everything else is the same. The workflow is the same. And once again, the taxa recorded from nearby sites are provided as a shortlist here to guide you in your uploads. All right. Then, if we wanted to add algae data to our site, that has a slightly different form that I think is just worth looking at. So everything's the same until we get to the broad biotope, which matches that of the invertebrates upload form, the same kinds of broad biotope options and specific biotope options. And sampling method is different. It's method specific to algal sampling, including things like scrubbing, collecting algae from the soft bottom of the stream bed, sampling from the vegetation, or an in situ collection. And sampling effort now includes replicates as well as time and area. And record type is the same, but curation process, you can specify that, whether it was acid cleaned, preserved in Lugol solution, or whether the sample was frozen. Once again, you can upload the site image. And algal biomass records are often biomass measures. And there are 2 options here on FBIS for indicating biomass. There's chlorophyll A and ash-free dry mass. For chlorophyll A, you can specify whether your indicator is chlorophyll AB, chlorophyll AW, and you can specify the sampling details, whether the whole cobble was sampled, whether it was just the sediment on the soft bottom of a stream or wetland that was sampled, an in situ sample, or whether artificial substrate was sampled. And then once again, the measurement can be indicated in milligrams per meter squared. Similarly, for ash-free dry mass, it will either be B or W, and the same sampling details are available here. Then, list of nearby algae taxa are provided. And just to point out that if you don't see the taxon here that you recorded, you can add it. And there's also even an option to add a new taxon if your taxon is not existent on FBIS as well. So just like for the other modules, you would indicate whether a taxon was observed at your site. And then here's another difference. You have some different options for algae in terms of indicating the abundance of algae in the sample, ranging from the species val or the frustule count, and percentage abundance, and then density as cells per meter squared or milligrams per, or cells per milliliter. All right, and then you would move on as per the other modules. Okie dokes. So now we've covered the fish upload form, invertebrate upload form, and the algae upload form. Just to show you the adult odonate upload form. Once again, very similar. And just to point out that the biotypes generally match those to, okay, these are not specified here. But these are generally match the standard invertebrate upload form. And sampling method is for odonates is a photographic record or a human observation or a visual observation. And record type as per the fish and invertebrates and algae dashboards. And once again, list of nearby adult odonate taxa and the option to indicate number, percentage abundance, or density. So basically identical to the standard invertebrate upload form, but this one specifically for adult dragonflies, which are a different module on their own on FBIS. And lastly is the anurin data upload form. If we found anurins at our site, this was the form that we would use to upload those data. And the broad biotype and specific biotype drop-down lists match those for invertebrates as well, as do the substrate lists. The sampling method has a drop-down including commonly used options for sampling frogs and tadpoles, everything from acoustic surveys to trapping and photographic records. The sampling effort could be specified once again by time or area, and the record type, once again, is the same as for other modules. And from there are no real differences from the fish or invertebrate upload forms. Your anurin taxa from nearby sites, whether or not you observed them, and how many you observed based on number, percentage abundance, or density. All right, so now that's really gives you a nice walkthrough of adding biodiversity data for the different modules on FBIS. And like I mentioned, in the next tutorial, we're going to be having a closer look at adding SAS data and associated data to an existing site. Thanks very much, and see you all next time.
Excellent. That hopefully gives you a good overview of the adding biodiversity data. So yeah, let's open it up for discussion. Questions, anyone? Please just raise your hand, or then we'll jump into the chat. Mark, welcome. Hi, how can we help?
Sorry, to turn my microphone on. Thank you very much. Okay, adding the species, you cover that one, Jeremy. Thanks for adding a new species or a species that might not be in the site already. So that's fine. My other question was around record checking. I know things, for example, like iSpot, if you are a novice, you get checked by someone else. Is there any way of checking the data, or do you assume the sampler has the requisite skills of identifying? Especially when it's visual records. When it's photographic or other records, you can get the data and double check. And then is there, second question, is there a recommendation to try and capture abiotic data? Because it's very important to link abiotic data to species occurrences, especially when you've got changes to the water quality. I'm looking at Gauteng with our water qualities here. And then the last question is, when you're adding a new reference, that was for, I think, the databases that you were showing, what if you make a mistake, you want to change it? You just add a new reference again, and they've got two of the same references. Is there any way of making sure that data is clean, so they don't have duplicates of the references, whether it's from River Health or something like that? Thanks.
Okay, thanks. Great, thanks, Mark. So just making a note. So the first one was about the validation. So we do encourage, so FBIS was kind of designed for storing of, you know, published reports and theses and what have you, rather than our naturalist type citizen science, you know, one photographic record, etc. So we make the assumption that if a student has done a master's or a report has been done for a consulting group, that there's some level of peer review and, you know, validation built in already. But that said, we have some validation systems built into FBIS. So when a user adds new data, it comes in as unvalidated. It is available on FBIS immediately, but it is categorized as unvalidated. And then the FBIS team will have a look at that data, and we've got some, for example, checks with, you know, fish species. We know certain species only occur in certain catchments. So if you add a fish species outside of its expected range, it won't be accepted, even at the adding stage. But then the admin team would go through the records and validate that these are accurate in terms of identifications. But you're right in saying, you know, without adding a photograph, we can't really say, well, that really was Pseudobarbus calidus, for example. The onus is on the person uploading the data, and a level of sort of training and understanding and peer review confirmation of IDs we are reliant on that. And then the same goes for the SAS validation. All data does go through a validation process, but short of being in the field with you, we can't validate it 100%. So there is a level of responsibility on the user uploading the data. And again, I reiterate, you know, FBIS is not really focusing on adding citizen science data. We rather direct you to our naturalist or the virtual museum for adding photographic records of fish, for example, which are authenticated, you know, and become research grade, and then that is pulled into FBIS. So the data will eventually be available, but they already have good systems in place for validating data, and we don't really want to, you know, repeat that here. Rather, let's rely on our naturalist and VM for doing that for us. So hopefully that, before I go on, does that adequately answer your validation question, Mark?
Yes, it does. Also, just for example, if you've had a species that maybe wasn't occurring there previously, that was outside its known geographic range, so would you be able to then just validate it if it's not part of that current list? So yeah, thank you very much. You've covered the topic.
Yes. So maybe just before I move on to the next question, I would like to just jump onto this testing site where if we go into just adding a new species, Jeremy did mention the sort of the workflow for adding taxa, obviously, but I thought it might be worth just to mention, if we go and want to add like if we get a species that's outside of our range. So say, for example, why is it not allowing me to add? There we go. So say I want to add a new species of fish, and as Jim mentioned, we use nearby records to kind of pre-populate this list. Now, if your species that you find isn't included in this list, you have two options. The one is to add a taxon, in which case you can just type in the first few letters. Say, let's just pick Pseudobarbus afer, You would observe it and add its abundance if you recorded it. When that gets added and you submit your data, that comes to the admin team who then authenticate it. And if pseudobiologus AFO was way outside of its range, it would be returned to you as, please check this. It's never been recorded in this area before. And obviously it depends a little bit on one's experience. I wouldn't sort of ask Paul Skelton to, question his records, for example. But yeah, so that's the built-in there. And then if you try to add a taxon, and I'm going to use a silly one here, I'm going to try and put my name and it didn't come up, it would mean that that name, Helen, is actually not on our master list, because behind the scenes, for every single species that we allow you to add, it's got to be in our master lists. So in this instance, the species Helen Dallas, because it obviously doesn't exist, isn't in our list, but you do have the option to add that new taxon and search for it. Now, if you know the species is an existing correct species, you could punch it in here. And it would link to GBIF and pick up the species on GBIF, because recall we use GBIF as our taxonomic backbone, and you could pick up, I know this is a non-native species, that's why I'm including it as an example, but you do have the option to add that. And again, I reiterate, the user can add it, but it still requires validation by the admin people. So if it's on GBIF, you would have the option to add new taxon and then select that taxon from GBIF. But if in my example, it's obviously a fictitious species and I ask it to find it, it's not going to pick it up on GBIF because it's obviously not a real species. And then you still have the option to add it, but you need to specify its rank and then add it. And that again comes back to us. And we won't allow it on the system until we validate it. Okay, so.
Thanks. Just a quick follow-up on the species question. With regards to the data, how do you cater for changes in taxonomy, either the splitting or lumping of species, with regards to the species data here?
Sorry, I didn't hear you. I don't know if is it your. I've got.
So can you hear me now?
Yeah, I can hear you. I'm just checking who's not muted.
Okay, I might have some background noise from the other people in the office.
Okay.
The other one is, what happens if there's taxonomical changes? In other words, a species is either split or the species is then grouped with other species through taxonomical research.
Yeah, that is obviously a difficult one, because historically it may not always be possible to split those species. I know there's a lot of new taxonomic work coming out of SCIB, especially with fish species. And if it's geographically delineated, like say, for example, some of the new galaxids that are going to be specific, or the Hierningness, the red fins that are very spatially delineated, then it would be possible to update those records going backwards. But it is a bit of a tricky one. I'm going to ask my colleague Jem just to jump in here, because I know he's been grappling with this a little bit because of the huge changes to a lot of the fish species names. Jem?
Yeah, thanks, Helen. And it's a really good question. Kind of one that I was hoping nobody would ask, but yeah, it is an important one. And I think you've covered it, Helen. It's going to be a case of trying to allocate. If you take a fish like the Galaxius, something that's been labeled as a single species that's in the process of being subdivided into multiple different species, some of which are spatially distinct. And in those cases, it's going to be straightforward to update the taxonomy and reassign the records confidently to the taxa. But in cases where you have spatial overlap, say these two lineages that were previously treated as a single species overlap in space, then it's going to be difficult to assign those accurately unless you have DNA samples or photographic samples or whatever's required to distinguish between them and go back and assign them correctly. So that's going to be a challenge, and I expect we're probably going to end up with a situation where some records are going to remain at a coarser taxonomic level, so that we, the first priority is the accuracy and the confidence in the data. So we may need to get a little bit creative there, but I think for the most part it's going to be doable in terms of the fish anyway.
Right, to come in there, Jeremy, I would assume if you've got the date record was taken and you can say this is the date before the taxonomic review and the date after, we'd be able to separate records that way as well. So that might be another aspect you can cover. But anyway, thanks for answering my question.
Yeah, that's a good point. Agreed.
Great. Thanks, Mark. Please, can you just remind me of your abiotic question so that I don't get it wrong?
No, I was thinking in Gauteng we've got a huge issue with the abiotic environment affecting species. So is there at least guideline or recommendation to collect abiotic data when possible? I know it's not always collected, but just so that it improves the ability to link species to abiotic influences, and especially when you start talking about government policies going forward. So how would use the data in this database in terms of making decisions. So that's my aspect coming to it from. I know abiotic data is not always collected and not everything can be collected, but is there a point to say to people, please collect as much as you can when you can?
I agree 100%. Yeah, I mean, we don't make those recommendations, but I think people generally know that, if possible and feasible within the constraints of the project, collect whatever abiotic data you can. And so being mindful of the importance of the abiotic data, that's why we actually built it in as a second form. You put your biological data, then as soon as you submit that, you go straight to an abiotic form, which hopefully, if you've got data, you'll add it then. So perfect, I agree with you. And then your last point was around the study references. Yes, you are able to edit those references. You do need to go back to the original site and then go to the source references and update them. At the moment, we don't have functionality to allow you in the back end of the source references, although it is part of the FBIS. If I just show you here, as an administrator, I can go into any source reference and I can search for anything that I have produced, and then I can go and edit this in the background. And I think in a way it might be useful to provide that functionality, because you don't always want to go through a million. Say you've added up 10 different sites with a number of biological records, all on pinned to one source reference, and then you find there's a mistake in that source reference. It would be easier to be able to just edit that specific source reference. For example, if I wanted to update this particular report, I could literally just go in, update, and say that was the wrong source. I could update it and then save it, and it's fairly straightforward. Whereas at the moment, a user kind of needs to go through the whole process. If I just quickly go here, I won't add in much stuff, but you go through the 2 pages, the biological record data, and then it takes you to the abiotic data. Let's just pretend we've got no abiotic data. Go to the last form. Oh, sorry, I hit the wrong thing. Go to the last one, and that opens up this source reference. Now, if you were to, in the next session, the SAS section, I do unpack this a little bit more detail in terms of this, but say you chose a database and you added a new one, tester database, and you gave it a description and you created it, and then you realized there was an issue. You would actually need to redo that, and we would have to fix it in the back end. So I think we need a way to allow users to update the source references themselves, the ones that they've created, because obviously you can't update other people's. So we will make a note of that, because I think it's a very good point. At the moment, you would have to redo it in this side. Okay, good. Thanks for those really useful questions, Mark. Hopefully that's provided some insight. Any other questions in the room? Any questions in the chat, Jeremy?
Nothing in the chat yet, Helen, but I'll just point out that we shared the links for the previous tutorials. It's a playlist on YouTube. So if you want to go and have a look, and if there's anything that you missed, you can watch that individual tutorial quite easily. Those links are there in the chat. I see a hands come up from Jane. Helen.
Okay, hi Jane, welcome.
Hi Helen, hi Jeremy. Can I just ask now exactly what is the role of a citizen scientist in this, or should I just be leaving it completely? Because, you know, I think I tried to submit some record and I got an email back saying that it's not for citizen science. So I haven't checked which one it was that wasn't accepted. But, you know, I think I'd tried to add abiotic data from our own tests and from other water testing records. So I'm not quite sure what it was that was rejected. But I just want to know if this is not for citizen scientists, then then obviously I'll just leave it completely and let you get all my records off GBIF. Which, you know, I've got a lot of stuff on iNaturalist, a lot of stuff then, you know, has made its way to GBIF. I know it's not all reflected, and I don't know if that's because the wetland module hasn't been fully developed yet, or if there's something, you know, it doesn't get updated that regularly. So just hear from you, what exactly is the role of the citizen scientist, and if it isn't aimed at citizen scientists, then it's fine. Then at least I know where I stand.
No, very, very good point. And thanks for raising it. So we 100% want the citizen science community to be part of FBIS, but we're sitting with the issue where a lot of citizen scientists do already add their data to either iNaturalist or to Virtual Museum, and any iNaturalist data that is sort of validated with a photographic record becomes research grade iNaturalist, as the same happens in the Virtual Museum. Now, those are two very well-established citizen science platforms. And then that data gets brought down to FBIS, but we need the master list. So, you know, not all, if the species isn't on our master list, then it's not brought down yet. So that is the proviso for that. So what we need to avoid doing in FBIS is for citizen science data to be added here as well, because then what we're doing is we're duplicating those records, which obviously is inaccurate and we want to avoid. So we took a decision quite early on in the FBIS system development to encourage, whilst we still want citizen scientists to use FBIS as a resource for extracting data, we're trying to encourage the adding of citizen science data to the existing platforms, which already have a validation process brought in, and which is then brought down to FBIS. So yeah, we definitely want you part of the community. It's just in terms of adding data, the workflow needs to go through the existing platforms, and then your data will then be brought down and available in FBIS. Happy to chat through that further if that doesn't answer your question adequately, or if you've got some other concerns, Jane.
Yeah, what is the abiotic data? Because we do our own water testing, you know, with our own instruments. I mean, you know, in the Jeremy's video, I talked about water clarity and that, and I think yes, well, you know, I do test with the Secchi disc, so you know, I've got that data. Yeah, and also I'm only referring to the one site that we look after, you know, our bird sanctuary, our wetland. So I'm not doing this like, you know, widespread. It's just the one site. And another, you know, so that's the abiotic stuff. You know, does my data have any value at all? And the other thing that I picked up in Jeremy's video was, you know, about the density of fish. Now, you know, we've been doing a removal of the invasive carp, you know, under permit from GDARD. And, we've now estimated the density per hectare. And, I'm wondering if does that type of data have a place or not? Is it just, because we obviously the carp is there as a species, but, we don't have the record of the density, which might be useful in future for, for people seeing how invasive it's become or whatever.
Yeah, I mean, I don't have any issue with you adding, abiotic data, to a site that's then monitored on a regular basis, because that's also like, standardized equipment. And, although you say it's under citizen science, it's not just, see what we can't do is just have the duplication is what worries us. So if you add your data routinely to our naturalist, then we probably don't want it added here as well. But if you don't have a home for your other data, such as abiotic data or fish abundance data, then FBIS can definitely, in my opinion, and Jem, feel free to jump in, it can definitely be a source for storing that data. And then what I would suggest is that you create a kind of a database or an unpublished data source that you then consistently use when you want to add your data, so it could be your organization's database. So we've created databases for a lot of organizations that routinely add data, such as Cape Nature has a SAS database. And then every time you add data, it gets allocated to that. And then, as you know, it's really then very easy to be able to find your data going forward. So say, for example, you added your data to, I'm just going to, obviously, if you were the owner of the data, that would be easy to find it under here, but you could also equally add it in as a specific study reference, which I'm going to use Cape Nature SAS data as an example here. Sorry, let me just. So you can see, that's not quite the right one, but anyway, let's just choose that for now. You would have a easy way to access, your organization's data on a regular basis. So hopefully that helps.
Well, maybe I can chat to you offline about that,
Yeah,
about creating a lot of that. Because like you say, I have no problem with uploading all my dragonflies and that to our naturalist and the virtual museum. But the abiotic data I definitely don't have a home for, and I think it's important to have it recorded. Not just here.
Yeah. Definitely.
Okay.
Reach out, and I'll set up a database, like a water quality database for you, and then that will be a great resource going forward. So thank you very much.
Okay, thanks, Helen.
Perfect. Okay, any more questions? Just wanted to check my notes. Yeah, so I think from our side that pretty much covers it. Any questions? Any more comments from your side, Jem?
No, not really. I just think it's an interesting discussion around the citizen science links, and the way I sort of think about it is, if you've got just a single observation, and typically a photographic record, then it's way easier to just send that through to iNaturalist, because it'll come down to FBIS automatically and will be accessible for all the sort of end use applications. But as soon as you doing a bit more of a quantitative sampling method, you're measuring your your sampling effort, and you're keeping track of your sampling methods, and you're collecting sort of more data points at a time, and or if you're collecting companion environmental data, I think those are generally the cases where you can capture more information by adding it to FBIS instead. And just to reiterate what you were saying, Helen, is just we're trying to avoid duplication as much as possible. The best place to avoid it is at the upload stage. So I guess for just users to make a call on what's going to be the most suitable platform and stick with the one. So, but it's an important conversation. It's one I think we're planning to have a focus session on next year with Nancy from Sanby to look at how these different platforms link together and to make sure it's really easy for the freshwater community, for everybody to see how these things fit together and to understand where and how the best workflows are for their data. So I think it's quite a big question and a very interesting one, and yeah, something to unpack more down the line. So thanks.
Agree 100%. It's not cast in stone, definitely, Jane, but yeah, we certainly value the role that you and your organization do, you know, so we don't want to lose that. And yeah, just keep the conversation going and help where we can.
Yeah, thanks very much.
Okay, Jem, no more questions in the chat at this stage?
Looks like that's it for today.
Okay, excellent. So once again, let me just jump onto my screen. We will be diving in the last session before we all have a much deserved break over the festive season. Well, not quite November, but December. We'll be diving into adding SAS data. Some of it, like the abiotic and the source reference, will be very similar to what we've done today, but we'll be adding in, you know, focusing on the SAS data capture side of things. So yeah, as always, thanks so much everybody for joining, and looking forward to seeing you again next week. Excellent. Go well.
Thank you.
Thanks, Mark.
Thank you, Jeremiah and Helen. Bye.
Thanks, Pumza. Go well.
Okay, thanks.